Good afternoon and welcome to the North Shore Outcomes Research Seminar and the 13th of our COVID-19 research series. As of today, we're about 2 years into this global pandemic that has claimed the lives of nearly 6 million people internationally and nearly 1 million people in the United States. But at the same time, we're seeing perhaps some bright spots on the horizon with over 700 million doses of vaccine in the arms of individuals around the world. And we're beginning to see a drop in the percentage of positive test results in the Chicagoland area and across the country. And yet we've been from peak to peak, and we really need an expert panel to help us better understand what lies in the future. And so I'm delighted to welcome our panel today and our guest speaker, Dr. Judd Holtquist, who will be joining us to present his research on the ongoing evolution of the SARS-CoV-2 pandemic, causes, consequences, and implications. This is in fact part 2 of a 2-part seminar series. This one is focusing on the Omicron variant. And we will also be joined by an expert panel, including Dr. Karen Call, who is the chair of pathology at North Shore University Health System, and Paige Larkin, Dr. Paige Larkin in microbiology from our North Shore. And we're going to be welcomed by Dr. Egan Ozer, infectious disease researcher and specialist from Northwestern. So I'd like to start off and once again thank all of the first responders and people working on the front lines in this pandemic. I'd like to thank the director of Outcomes Research, Dr. Bernard Ewickman, as well as all the program directors in Outcomes Research and the North Shore leadership for sponsoring this seminar series, and for the ongoing support to make the entire team work in fighting this pandemic. And I'd like to introduce Rachel Brewington, who is my partner in this process, and she is a facilitator for this seminar. Rachel will be working with me and to help facilitate the panel as we go through the seminar today.
Thank you so much, and welcome everyone. In today's webinar, all attendees will be muted with the exception of our panelists and speakers. However, there will be some opportunity for interaction. If you have any questions or comments at any point during the presentation or the Incubator Hub discussion, please submit your communication by using the chat function as noted by the blue arrow on the screen. If you are on a computer, you can access this icon by hovering your mouse over the bottom of your Zoom screen. Please give special attention to the audience of your message before you submit it. As you can see on the image to the right, you can choose to message either the panelists and speakers, this will include myself, or you can choose to message everyone on the webinar. Feel free to leave your questions and comments at any point during the presentation. If you are experiencing any technical difficulties, please let me know by messaging all panelists and I will assist as best I can. Okay, thank you so much. And Dr. David, take it away.
All right, thank you very much, Rachel. So I'd like to go over the agenda for the seminar. After our introduction today, we will have a presentation from Dr. Holtquist from 3:10 to 3:40. We may actually get to this presentation a little before 3:10. This will be followed by a question and answer session that runs from 3:40 to 3:50. I encourage you throughout the talk, as you think of questions, to be free to include those in the comments, and we will keep track of those and we will feed those to the speakers and the panelists. Following that, we'll have our Incubator Hub expert panel discussion where I will introduce each of the panel members as well, and we're going to go through some discussion questions to talk about the implications of this research for health equity, for clinical implementation, for policy, and translational science. And then we will have some closing remarks where we can ask you to fill out an evaluation, and we'll direct you to our YouTube channel where you can access this for some of your own learners who may have missed this seminar today. Very good. So I would like to introduce our guest speaker, Dr. Judd Holtquist, who is assistant professor at the Northwestern University Feinberg School of Medicine and Infectious Diseases. He's also associate director of the Northwestern Center for Pathogen Genomics and Microbial evolution. Dr. Hellquist is also a collaborator with several of us on this call today, and we are just delighted to welcome him back to the Outcomes Research Seminar for his presentation. And I believe he just gave grand rounds at Northwestern this morning, so he has been very busy. So welcome, Judd. Thank you so much for joining us today, and we're going to allow you to share screen so you can drive the talk.
Fantastic. Thank you so much for the intro. Let's see. You can see everything clear? Beautiful.
Yeah. Oh, actually, it's a little pixel. I think you need to do slideshow. Maybe it's just me.
Let me know, and I'm happy to switch over to an alternate format if we need. But thank you again for the invite to come back for part 2 of this series on the ongoing evolution of SARS-CoV-2. You know, when I gave the part 1 in October of last year, I think I had said the word Omicron maybe 100 times in my life in total. And now I'm going to probably say it about 100 times in the next half hour. But it just goes to show how quickly this pandemic continues to move and why it's so important to keep track of. So just to give everybody kind of an update on what I talked about last time I had discussed how we use cellular and molecular assays in the lab to better complement genomic surveillance strategies and inform the causes and consequences of SARS-CoV-2 mutation. And in this part of the talk, what I really want to focus on is how those mutations are impacting the viral spike protein and what the new Omicron variant ends up looking like.
Sorry, could you switch to slideshow? I don't think it's coming through. Thank you, very good.
How is that?
Perfect. Okay, you're on. Great.
Beautiful. Quick disclosures, we get research support from Gilead and do some consulting for Blue Cat Bio and Horizon Discovery. So just to get us started, you know, viral mutation is a natural phenomenon. All viruses mutate. And those mutations really come from 2 primary sources. There are intrinsic mutations and extrinsic mutations. So intrinsic mutations are those that arise during replication of the virus due to errors made by the polymerase. Extrinsic mutations are mutations that arise due to some sort of external source. So this can be natural deamination of a base, it can be due to NTP imbalances in the cell, or it could be due to mutations induced by a viral enzyme or a host enzyme like the ApoBec family or the ADAR family of enzymes. But at any rate, all viruses mutate. Now, lucky for us, SARS-CoV-2 has a relatively low mutation rate. So if we look at the SARS-CoV-2 on our this chart of mutation rate on the y-axis and genome size on the x, we see that SARS-CoV-2 actually has among the lowest mutation rate of all known RNA viruses. And this is actually for 2 primary reasons. One, SARS-CoV-2 also has among the largest genome of any known RNA virus that infects humans, coming in at a little over 30 kb, but it also encodes for its own proofreader enzyme that goes and checks that each base pair laid down by the NSP12 polymerase is indeed correct. And both of these factors help keep the mutation rate of SARS-CoV-2 down fairly low. But that being said, it is still mutating, just not quite as much as we might think of if we have other virologists in the audience. So I absolutely love this slide, and I think it shows the point really beautifully. This was created by a collaborator in the Center for Pathogenomics, Dr. Ramon Lorenzo Redondo, but it's showing the total degree of diversity we see in one gene of HIV, this is HIV polymerase, over a 1-year period in Chicago, compared to the entire diversity in the SARS-CoV-2 genome over the entire first 2 years of the pandemic. So we can zoom in on that a little bit more, and you can see that there is diversification of this viral family, but when you compare it to some other RNA viruses that we're familiar with, the mutation rate is quite low. But SARS-CoV-2 is diversifying. And this is critically important because genetic diversity is the basis for natural selection. This is the exact same concept that we all learned in primary school. But you have a population of viruses, mutation ends up creating variation in that population, and unfavorable mutations get selected against. But over time, you're going to get a bigger and more diverse pool. Now, what happens in cases where you have some sort of selective pressure on that population? Favorable mutations are more likely to survive, reproduce, and continue to grow. So we usually learn this when we were in elementary school using some example with beetles or birds or flies, but the same thing actually applies to viruses. And there are multiple selective pressures that act on viral populations. They can change in order to transmit better, either between different species and humans or between different humans. But they also have selective pressure to avoid immune responses and antiviral drugs. So at the end of the day, it ends up being critically important that we're able to track SARS-CoV-2 mutations and also determine which ones might be harmful. So there are three basic things that we really need to do to identify which mutations we need to be concerned about and which ones might just have arisen naturally and are not of dire consequence. So the three things that we need to essentially be able to do is, one, we need to be able to identify mutations in real time. Number two, we need to be able to associate those mutations back to clinical data to see if they change either presentation, clinical outcome, or therapeutic effectiveness. But then three, we also need to be able to establish causation using a series of lab-based approaches. So very early on in the epidemic, my colleagues in the Center for Pathogen Genomics got together and we decided that we needed to set up a system for tracking ongoing viral mutation in the city of Chicago. And this is how our current infrastructure works. So we have partnerships now with Northwestern Memorial Hospital and Lurie's Children's Hospital. And essentially any residual diagnostic specimen that was tested for SARS-CoV-2 that is positive, and this can come from outpatients, inpatients, healthcare workers, the drive-through clinic, pre-surgery, et cetera, ends up getting routed to our lab space. We then take those samples, de-identify them, and put them in our biobank repository. And we just banked over 22,000 sample earlier last week. Those samples are then sent for RNA extraction on our automated robot. We can quantify viral load using qPCR, reverse transcribe the RNA to DNA, and then sequence the viral genomes on the Illumina MiSeq platform. So of our 22,000 positive samples that we have banked, we've so far sequenced over 4,000 of them, and we try to, and as close to real time as possible, deposit those sequence in the online repository GISAID. Now, what's really cool is we're doing this all in the context of a single hospital system. So what that enables us to do is take those genetic data and relate it back to the clinical and demographic data that we have available within the hospital medical records. So this slide is essentially trying to take all of the sequencing data that we have done over the last couple of years and compress it into one simple schematic. So let me walk you through it quick. So on top here, what we're looking at is a graph of daily case counts over time in Chicago from March 2020 to January of 2022, and that's in this yellow line here. We're also overlaying that with hospital admissions in blue, deaths in red, and then the percent of fully vaccinated individuals in this kind of sage green line here. And what we can see when we trace this out is really the pandemic in Chicago happened in 5 distinct waves. Wave 1, wave 2, wave 3, wave 4, wave 5. Now, what's interesting to note is that each one of these waves has been associated with this unique blend of variants. So below here, we're plotting out the cumulative distribution of variants that we had sequenced in the city, going all the way from the original Wuhan strain in this very light blue to the most recent strains of Omicron. So the very first wave, and this was very early in the spring of 2020, the epidemic in Chicago was primarily consisted of 3 different clades, a blend of clades 20A, B, and C that outcompeted the original Wuhan virus. This was then supplanted by regionally specific variants in the fall of 2020. And here in Chicago, that variant was called 20G. In the spring then of 2021, variants of concern, or VOCs, came to dominate. And here it primarily was made up of alpha and gamma. This was then displaced by Delta over the late summer, early fall of 2021. And then, of course, at the end of the year and into the start of 2022, we saw Omicron sweep the globe, and that led to the highest level of case counts that we saw in the city to date. Now, as we progressed from 20 ABC to 20G to alpha gamma to delta to Omicron, we saw these waves continue to sweep through the city. And what's important to understand is that each new variant carried a unique selective advantage that enabled its emergence. Now, there are many different reasons why these variants have been optimized over time, but a lot of the reason comes down to changes in the viral spike protein. So I really want to focus in on SARS-CoV-2 spike for the rest of this talk until we get to Omicron. So SARS-CoV-2 spike is this outer protein on the outside of the virus, and it's really important for two primary reasons. One, it's essential for viral entry. So it binds to the receptor ACE2 and facilitates entry of the virus into the host cell. But the second reason that spike is really important is because it is the primary protein on the outside of the virus that's recognized by host antibodies. So if you have an antibody response to SARS-CoV-2, exactly what part of the spike protein is recognized is really important in order to launch our host cell defenses. Now, the spike protein, because it is so important for both transmission and for immune evasion, has been changing rapidly over the course of this epidemic. And that's what we're showing in this heat map here below. So this very top line here represents mutations going across. And this top line is this first 20 ABC clade. The darker the purple, the more frequent the mutation. So we see in this entire first clade, there was one dominant mutation, and that was it. Now compare that over time as we went to 20G, to the variants of concern, and then especially to Omicron as we got more and more variants accumulating in the spike protein. So in order to understand exactly why these mutations were essential, we need to understand a little bit more about spike. So I'm going to take a slight detour in order to explain exactly how spike mediates entry into the host cell. So the first thing to know is that when you are producing new viruses, the spike protein is actually cleaved into two parts. So when you have a viral genome in the cell, the virus buds into the endoplasmic reticulum, and the spike protein in the ER is cleaved by host proteases known as furins. Now, when furin cleaves the spike protein, and it happens right here at this subunit 1, subunit 2 boundary, we end up getting a structure that finally folds like this, shown here in the blue and the red. So the final spike protein kind of looks like a mushroom, where you have S2, which is the stalk, that's planted into the virus membrane. And then you have the S1 subunit, which is the head of that mushroom. And that sits on the top, and that's what's responsible for engaging with the host cell receptor. Now, once this final fold is formed, we get this nice closed structure where we have 3 different spike proteins that form a trimer. And when we look on top of it, we have the N-terminal domain, and then we have the receptor binding domain that's laying flat on the top of this spike. Now, that closed structure actually cannot bind to the host receptor ACE2. Instead, what actually needs to happen is you have to see it have a conformational change in spike in order for the receptor binding domain to bind the receptor. So what happens is actually these N-terminal domains have to slightly shift, and that shift flips up the receptor binding domain, and then the receptor binding domain can bind to ACE2. So this conformational change is really important for actually engaging the receptor and infecting the cell. Now, after ACE2 has been engaged, then we get the action of the co-receptor TMPRSS2. Now, once ACE2 is bound, this enzyme can come in and it essentially cleaves the spike protein for a second time. That occurs just downstream of this original cleavage right here. And when this part is cleaved off, two things happen in very quick succession. First, this outer cap of the mushroom, this S1 subunits, gets shed. And we get this really rapid conformational change that essentially skewers the host cell membrane with the fusion peptide of the spike protein. Now that skewering essentially now has linked the two membranes together. And that is all initiated by this co-receptor and cleavage enzyme TMPRSS2. Now, once that other membrane is skewered, we begin to get a folding back of this protein on itself, where you see these little loops here. They actually begin to form coiled coils, and these coiled coils are incredibly structurally stable. And so they're they provide the energy by which these two membranes come in close proximity and eventually fuse, enabling the viral genome to enter the host cell. The spike protein of SARS-CoV-2 is absolutely an amazing structure. The protein and the mechanics and the work going in to understand it has been phenomenal. And understanding exactly how it works is important for understanding why these mutations are so impactful. So now let's rewind again. Let's go back to those first two waves of the Chicago pandemic. So those two waves were actually caused by a single mutation in the spike protein. And that mutation is known as D614G. Now this mutation ended up being really, really important for two primary reasons. One, that mutation causes this top upper subunit, S1, to bind better to S2. Without that mutation, what would happen often is the S1 part of the protein would simply fall off when the virus was being produced, making more uninfectious viruses. And this mutation enabled more infectious trimers to actually be produced. Now, the second thing that mutation does is it actually enables this closed conformation to switch to an open state more frequently. So that means essentially you have more opportunities for the spike protein to engage the ACE2 receptor. So that single mutation ended up really being essential for this virus to transmit easily in humans. Now, the one issue with this open conformation is, the question I get asked all the time is, well, if that's true, why wasn't the spike protein just always in that open conformation? Why wouldn't you always be prepared to engage the ACE2 receptor? And actually, this is because that same binding site is highly susceptible to neutralizing antibodies. So most viruses actually have their spike protein in a closed state and only rarely flip it open. Because in this open state, you can have antibodies binding here, and it'll completely prevent that spike from being able to enter the host cell. And once this new open state was being accessed more due to the D614G mutation, we saw more and more neutralizing antibodies being formed. And when we actually map where these neutralizing antibodies are binding, they all bind to this upper surface, and most of them bind to this receptor binding domain. So it's probably no surprise that shortly after that time, we saw the emergence of all of these new variants that all had mutations to resist neutralizing antibodies, and these mutations were heavily biased towards the receptor binding domain. So now let's fast forward to this wave 3 and 4. These are the variants of concern, and these variants of concern had these two properties. They had enhanced transmissibility, and they also had decreased susceptibility to neutralizing antibodies due to all of these mutations that had accumulated in their receptor binding domain. Now, what was really interesting about these variants of concern is that we saw several different variants that all had the same mutations, but all of these mutations actually arose independently from one another in an instance of convergent evolution. So a few of those mutations are highlighted here. So for example, beta and gamma both shared an E484K mutation, whereas the delta and the alpha variant both shared a mutation at P681, etc. So what was really interesting at this part of the pandemic was we saw all of these variants of interest and all of these variants of concern emerging throughout the world in different locations in a very narrow time frame at the end of 2020. So the question quickly became, where were these variants coming from? And so we partnered with some clinicians here at Northwestern Medicine to perform a longitudinal sampling of people who were infected with SARS-CoV-2 in our hospital. The idea being that perhaps SARS-CoV-2 is diversifying within patients, and these accumulated mutations can lead to the production of these sort of variants or mutations of concern. So we sampled patients who were not in the ICU, patients who were in the ICU, and unfortunately some patients who ended up passing away. And we sequenced their virus at various times over the course of their infection. Now, when we looked at what viruses these patients were carrying, we actually very rarely saw the virus change from its original state. But in 9 out of the 26 patients where we were able to successfully sequence, we did see a single or maybe 2 mutations pop up. But in only 1 case out of all these patients did we see a mutation arise in the spike protein. The reason for this is that patients in the hospital typically can clear the virus quick enough that it doesn't have time to accumulate the mutations necessary to really be selected for. Now, at this point, we were approached by several different clinicians in our hospital who said, well, what about in the case of persistent infection? So, for example, in immunosuppressed patients who can't clear the virus and are seeing the virus continue to replicate for much longer periods of time. So we actually partnered up with a couple of clinicians here at NM to collect longitudinal samples from a few immunosuppressed patients in our clinic. Here we got 2 different patients, patient A and patient B, and we were able to actually sequence their virus over 100 days apart in both instances. Now, what happened when we looked at the mutations in these patients is we saw that the amount of viral diversity in these patients increased at every single time point that we ran a sequence. To by the end, we saw a massive amount of diversity within the virus in this 1 single patient. And when we looked at the spike protein in these 2 patients, here we're looking at the spike protein at 8 days post-infection, 53 days post-infection, 94, 95, and 131 days post-infection. You can see the accumulation of these mutations in the spike protein in this patient. And the same thing was true for the other patient. And when we compare the mutations that arose in these spike proteins, several of them were actually shared with known variants of concern. So patient A actually evolved mutations that were shared with the beta variant, and patient B evolved some mutations that were conserved with the gamma variant. So that made us think that perhaps these immunosuppressed patients or patients who have persistent infection are one source of some of these concerning variants that can evade neutralizing antibodies. So we thought that we had a pretty good handle on how the virus was mutating. And then, of course, we all got the massive surprise of Omicron. So Omicron really has distinguished itself in how heavily diversified it is relative to the previous variants of concern. So again, now looking at this map, again, you can really appreciate just how many mutations are in the spike protein. And they are highlighted again here, and you can see a little bit more clearly in this figure below how they're really clustered in the N-terminal domain and the receptor binding domain of the spike protein. It's really hard to understate how mutated this virus is. So this is one of my favorite graphics that I think does a really nice job illustrating this, is this looking at viral sequences over the course of time. And we're plotting mutations in the S1 part of the spike protein on the y-axis. Then these different colors are different variants of concern. So for example, alpha generally had about 6 mutations per spike protein. Beta had a little more than that. Delta, we had somewhere between 5 and 10 in general. And now look at Omicron, upwards of 20, 25, up to 30 different mutations in the S1 subunit of spike alone. So just a massive number of changes. And this divergence of Omicron spike has real world implications. The first of all being that it severely has limited the effectiveness of several monoclonal antibody therapeutics. So some of the variants of concern that we had talked about before have mutations in the receptor binding domain that had specific impacts on specific therapeutics. But with Omicron, there are so many mutations clustered in this receptor binding domain that it impacts the ability of several of these monoclonal therapies to truly be effective. So for example, I'm just showing a smattering of them here from this recent paper by Van Blargen in Nature Medicine. But what they were showing here in black is the D614G spike, and in blue is Omicron spike. They were adding these neutralizing antibodies up top and then looking at how effective the resultant spike proteins were. And what they were able to show is that these black D614G spikes were neutralized by each and every one of these neutralizing antibodies, but Omicron was resistant to a large majority of them. Now, the second major thing that this large divergence ends up leading to is it decreases the neutralization activity of antibodies produced following vaccination. So neutralizing activity was severely reduced when you compare Omicron compared to the Wuhan strain and when you compare it to Delta. So just to briefly run you through some of the figures from this paper, which was fantastic. It's Garcia Beltran 2022 in Cell. Definitely give it a read because it's really, really interesting. But here what we're looking at is we're looking at serum that was isolated from people who were vaccinated with the Moderna vaccine either twice or 3 times, with the Pfizer vaccine twice or 3 times, or with an adenovirus vaccine followed by a Moderna shot afterwards. And we're comparing now a D614G virus, a Delta virus, and an Omicron virus. And what was fascinating is that you can see that when you go from 2 vaccinations to a booster, you get more neutralizing activity against wild type. That same is true for Delta. But when you look at Omicron, you can see that these two vaccinations alone do not provide that much protection, whereas this booster was actually massively helpful. And this is with Moderna. You see the same difference with Pfizer. So the neutralization activity of antibodies produced following vaccination was much lower. And that leads to the obvious consequence that Omicron has been associated now with decreased vaccine efficacy. And right now, these studies are just beginning to come out. So I want to highlight 2 of them that I think are pretty notable. The first one came out of South Africa, where they reported that 2 doses of the Pfizer vaccine really had an efficacy of only 33% in preventing infections. Now, that is presenting infections, but most of the people who actually did get infected with Omicron and were vaccinated had very mild disease. And so the vaccine was still 70% effective at preventing hospitalization. Now compare that to over 90% effectiveness in preventing hospitalization for Delta. Now a very similar study had recently been reported that compared 2 doses relative to 3 doses, again comparing Delta versus Omicron. And in this case, they found that Omicron led to more hospitalizations with 2 doses, where the 2 doses were 81% effective at preventing hospitalization against Delta, but only 57% in preventing hospitalization from Omicron. This could largely be mitigated by the addition of a booster, where it was about 90% effective against both of these variants. The last thing that I kind of want to note about Omicron, and could be a result of the spike protein differences, but we can't be sure, is that Omicron has been presenting with a somewhat altered symptom profile. Now, while several of the symptoms have been conserved, there are a few of which have changed most dramatically. So for example, with Omicron, there's much less likelihood that patients are presenting with a loss of smell when you compare that to Delta. So going back now to the evolution of Omicron itself, it's really curious and shocking to a lot of virologists, and myself included, that we entirely anticipated the next variant to be evolved from Delta. So prior to the emergence of Omicron, Delta had made up over 98% of circulating sequenced variants that were being reported. But when we sequenced Omicron, it actually is derived from a completely independent branch of SARS-CoV-2 evolution. So here is Delta up here in this lightish blue, and here is Omicron here in that red. And this actually has really important implications on what the future pandemic might look like. So for example, H1N1, since it was the pandemic in H1N1 jumped over in 2009, the evolution of the virus has followed a fairly straight pattern. This is known as an imbalanced or a ladder tree, where whatever the most successful variant is, gives rise to the next variant, gives rise to the next variant, gives rise to the next variant. That means that we have case numbers low enough and we're driving evolution in one directed manner through our vaccines and our public health measures. With SARS-CoV-2, however, there's no clear evidence that that's happening yet. And what's really interesting is if we compare another endemic coronavirus, such as CoV-OC43, we see two distinct branches that form over long periods of time. So this is sequencing done over decades. So what we don't know yet is if SARS-CoV-2 is going to follow a more ladder-like pattern like flu, or if we're going to actually get two sublineages that become dominate and perhaps alternate over time. But the main question then that people keep coming back to is, where did Omicron come from? And really now there are three main theories that are being discussed. One, Omicron arose after long-term selection intra-host. So this would be similar to that persistent infection profile I had talked about with the emergence of variants of concern. While that idea is appealing, especially seeing how many changes are occurring in the receptor binding domain, this would require an extremely long and persistent infection, and truly has been unprecedented in anything else that we had sequenced from these patients in the past. So the second idea is that it could have evolved in a population that's just not regularly sampled for surveillance. So when we look over the entire globe, there are many regions that are severely undersampled, and it could be that maybe it emerged quietly in one of these regions, became more infectious, and then broke out. That is a possibility, but again, it's unlikely that we never would have seen anything like it before. So the third theory is that perhaps Omicron is a product of interspecies jumping. So it jumped into humans, jumped back into a different species, and then came back to us. And again, this is another appealing idea, but it's a very difficult thing to prove. And most likely, it could be a product of one or more of these different reasons combined. And the most interesting thing perhaps about Omicron, from my perspective, is it's not done yet. So it's continuing to evolve and change over time. And in fact, there are multiple different sublineages of Omicron that have cropped up since major Omicron has arrived itself. Most notably, I want to point out this BA2 sublineage, which is right about here. And when we look at the mutations that BA2 shares, it clearly is a derivative of Omicron, but then it has several mutations that are actually quite distinct from the main lineages of Omicron, BA1 and BA1.1. And this has actually led to the concept that perhaps SARS-CoV-2 is recombining in patients. So if you look at this half of the spike protein of BA2, it's very similar to BA1. And you look at this half, it's not very similar at all. And so there's this idea that perhaps the genome from one and the genome of another could have recombined to create this hybrid strain. Now, this requires that somebody is infected with two different strains at once, and that's a very difficult thing to prove, but something that a lot of researchers are actively focusing on now. So BA2 is circulating in Chicago. So this is their most recent sequencing data as of the very end of January. BA2 is present, that's this tiny little bar here, but not quite frequent yet. So just to kind of sum up what we talked about, each new variant has carried a unique selective advantage that's enabled its emergence. And while these mutations and the rate of change might be surprising, the overall trend really is not. So in 2020, we saw the emergence of these strains that had enhanced transmissibility. In 2021, we saw a coupling of enhanced transmissibility and limited immune escape. And then in 2022, we have the combination of enhanced transmissibility, broader immune escape, and now some evidence of vaccine breakthrough. So it doesn't take too much imagination to predict perhaps where the next evolutionary trends are going to be going. So with that, there's still a lot of open questions that are left to answer. There's a lot of talk about, is Omicron less severe, or is that just a perception that we have due to increased breakthrough infections and a better protected population? There's still the outstanding question of where Omicron came from and what that could mean for the emergence of other highly mutated variants. There's also the very large mystery of what happens during the winter season when we see these surges. So for example, here in Chicago, we saw this huge escalation in 20G cases, but there were no corresponding changes in the spike protein. So was this a founder effect, or are there other changes in the genome that are driving significant impact in human infection? Fourth, what's the periodicity of this variant going to end up being? So if you remember back from my last slide, it seems that every 4 to 5 months, we're seeing a peak here in Chicago and a unique variant strain. Is that pattern going to continue with a big winter surge and 2 minor peaks, one in the spring, one in the fall? And then finally, how much longer are current vaccine formulations going to remain effective, and how often are we going to have to update those to stay on top of this going forward? So with that, I just wanted to, shout out a huge credit to my colleagues in the Center for Pathogen Genomics and Microbial Evolution, Ramon Lorenzo-Redondo, Egan Ozer, who's actually going to be joining us on our panel, and our lab coordinator, Lacey Simons, who has personally sequenced so many of these viruses. It's crazy. Huge credit to all of them. A lot of our collaborators, including Chad Achenbach, who did a ton of work in helping us to gain access to a lot of the EDW data. Our colleagues here at Northwestern and in the core, as well as with their funding sources. And with that, I would be very happy to take questions.
Thank you very much, Dr. Hellquist. And I'm going to pass this over to Rachel, who is going to field the questions. I can start off in the meantime. And we had a question from Stephen Wachtel about whether this is Darwin's theories in action, the evolution of the virus. Whether you could comment on that. And now that you're, Rachel, you can take over.
Yeah, and while you're transitioning to Rachel, absolutely. And it's viral evolution is just like the evolution of anything else, but we're seeing it in such real time, and we're seeing it evolve rapidly. And part of the reason that we can observe it, really evolving in real time is there are so many cases and so many viruses. That enables pathogens to sample an amazing amount of genetic space in a really short period of time. So if we're thinking about mammalian evolution, we have to think over the period of millions of years. And in viruses, you can do these kind of experiments in a test tube. And we're watching a real life experiment go on in real life as we have unvaccinated people mixing with vaccinated people, and we have different people with different levels of protection and administering different drugs. And I probably shouldn't be nerding out so much about SARS-CoV-2, but from a virologist perspective, it's been fascinating.
Okay, there's another question by Jane T. Is there data on people who get infected with Omicron, for example, with a J&J single vaccine, having subsequent protection from getting reinfected?
I'm not particularly acquainted with the J&J information. It is recommended that J&J shots have been followed up with an mRNA booster, but I have not seen how those fare against Omicron.
Thank you. Okay, this question is from Sarah Polanski. How have the current variants in Omicron affected ability of tests to detect it?
How the variation can impact tests? It's a great question. So there are a couple different tests that we have to be concerned about. The first are our PCR-based tests. So the PCR-based tests require binding to specific sequences in the genome. And if there's a mutation at one of the sites that these probes are binding to, it can cause for false negative tests. Now, most of the assays have been designed so that they avoid regions that are heavily mutating. But a great example of this is actually what's known as the S gene dropout. So there are diagnostic tests that actually had a probe over a site in the spike protein that was changing in some variants. Now that was causing the S product to actually disappear from some of these diagnostic tests. But nicely, we always have to think ahead about these things. And these tests are actually multiplexed, by which I mean they were probing for multiple different parts of the virus at the same time. So those samples still were coming up with positive. They were just missing that one S product, S gene dropout. So it is something we have to be concerned about, and we have to continue to assess, as the pandemic continues to evolve.
Thank you. This question is from Dr. David. Is there evidence of Mueller's ratchet in the evolution of SARS-CoV-2?
Excuse me?
Muller's ratchet? Is there evidence of Muller's ratchet in the evolution of SARS-CoV-2?
I should probably be familiar, but I'm not.
Oh, it's a phenomenon of deleterious mutations making the virus less virulent over time. Is that?
Gotcha. So in terms of the evidence that viruses naturally tend to become less virulent over time, there is, I would say, mixed evidence to that theory, because really we can't think of pathogens just by themselves. Whenever you're thinking about an infectious disease, you're really thinking about a bipartite system. There's the pathogen and there's the host. So what the pathogen does is really important, but how protected we are and how we respond to pathogens is equally as important. So for example, with SARS-CoV-2, one big question about the Omicron variant is it truly less severe, or is it just replicating in a population that's better protected against it due to a combination of prior infection and vaccination?
Thank you. Okay,
Thank
this
you.
question is from Dr. Call. Do we have any information yet on polymorphisms in the ACE2 and susceptibility or severity of disease?
That's a great question. And I need to do some more literature diving on that. So the question boils down to, there are known human genetic differences where the receptor, that ACE2 protein, can be slightly different person to person. And the question is, do those differences also influence how well SARS-CoV-2 can infect someone, or what cells can actually be infected? And I'm actually not sure what the answer is, but it is an incredibly interesting area of research. There has been reports about how small changes in immune genes can influence severity of disease. So for example, if somebody is defective in an immune response to the virus, they get more severe disease than others. But in terms of the receptor, I'm not sure.
Thank you. And our last question from KN to everyone, can this same type of genomic analysis be applied to understand how SARS-CoV-2 evolved in nature? What is the reservoir species for SARS-CoV-2 and where can it be found? Is there a missing intermediate from RATG13?
Yeah, it's a great question. So what we do know is that there are similar beta coronaviruses in bats. And the most similar virus that we've been sure to find is in a bat. Now, when you look at how different that bat virus is from SARS-CoV-2, the question, most people believe there had to have been an intermediary. For a while, we thought that we knew which species that was, and now that's being more debated. And at the end of the day, I think we still aren't exactly sure. Now, this is probably a conversation for a different day, but we have trouble even funding our own people to sequence human samples. Funding for animal reservoirs has been woefully depressed. And so it's securing more funding to do more surveillance in animal communities, we think is going to be really important. And that's especially the case now that we're seeing this heavily mutated Omicron arise, that if we can get reverse transmission from human back to other species and back to us, that means we really need to begin paying attention to these potential zoonotic reservoirs. But it's a fantastic question. Genomic surveillance in general is just so important. In the center, we're trying to do the same type of analysis to other pathogens as well, other viruses such as RSV, but then also to resistant bacteria, antimicrobial resistant bacteria, and anti-fungal resistant fungi. So really the approach is really applicable. We just have to be better at applying it in force.
Thank you so much, very much. Okay, that is all the time that we have for questions right now. Please feel free to continue to insert any additional questions in the chat box for our presenter to review later, and they'll get back to you. I will now hand it over to Dr. David, who will introduce our expert panelists for the Incubator Hub panel discussion.
Thank you, Rachel, very much. And thank you for those really fantastic questions. And Dr. Hultquist, again, thank you for a brilliant presentation. It's just absolutely fascinating. And so encourage participants to send in comments for the panel. But now I would like to introduce our three expert panelists who will be joining Dr. Hultquist to provide some additional insight into the implications of the research presented. So today we have Dr. Karen Call, who is chair of the Department of Pathology and Laboratory Medicine and the Duckworth Family Chair at North Shore University Health System. And she's also a clinical professor of pathology at the University of Chicago. I'd also like to introduce Dr. Paige Larkin, who is Director of Molecular Microbiology and Associate Director of Clinical Microbiology at North Shore. She's also a clinical assistant professor at the University of Chicago. And we're also joined by Dr. Egan Ozer, who is a microbiologist, and he's an infectious disease physician and director of the Center for Pathogen Genomics and Microbial Evolution at the Havey Institute for Global Health at Northwestern. So welcome and thank you very much, panelists, for joining us today. Next slide. So for our expert panel Incubator Hub discussion, I'd like to ask the panelists to discuss the implications of this research first for health equity. So based on what you've seen here and based on your observation of access to testing, treatments, vaccinations, a range of topics, please share your thoughts about the health equity implications of this research.
Yeah, I guess I'll start. And thanks again, Judd, for a wonderful summary of what we know about Omicron and really sort of our state of knowledge about the pandemic itself from a genomic standpoint. I would really like to see all that data together in one place. And so thanks again for putting it together. And thank you again also for inviting me to this Incubator Hub to discuss these topics with Judd and Paige and Karen. So, but I think the things that jump out most to me from, I'll just start with one and we can talk about others, but the thing that jumps out in terms of health equity here in the United States, and we can talk about global as well, is that, Judd's data really, and that he presented really showed how important the booster shots are to protection against Omicron. And we can, sort of interpolate that that's going to remain an important factor for protection against potentially future variants of the virus that are likely coming down the pike at some point. And so we really do need to, I think, do a better job of distributing vaccines, and not just the 2-dose vaccine and done, but really making it clear that the third dose is beneficial in protecting against infection and severe disease, and making sure that there is broad access to vaccinations within all populations.
Thank you very much. Thoughts from others on the health equity implications and maybe expounding on what Dr. Ozer just discussed.
I'll chime in, and again, thank you for including us all today. I think it was a brilliant, summary of where we're at, Judd. And it's always wonderful to hear you, bring us up to date on what's going on. So I learned a great deal and have enjoyed it thus far. It excites me and it frustrates me to see where we're at here 2 years down the road, because we're still having some of the same discussions that we've been having. We know some of the lessons we should have learned, and we still haven't been able to implement them. So as Egan mentioned, we're still not where we need to be with vaccinations, and we're still having that battle. And, it's very clear, even though the virus is evolving and we are, I think, likely to have new vaccines down the road to keep up with new variants at some point in the future, we're still not where we need to be with the, existing, 3 vaccines that everyone should be getting. And so that's frustrating. The testing remains an issue. There's a lot of confusion out there about, when to test and what to use and how to interpret the findings. And I think, we still are not where we need to be with that. Hopefully at some point, we'll reach a scenario that's like flu, where, we are all getting vaccinated with an appropriate vaccine, and we can test only those people who are symptomatic. That may be a pipe dream, but I'd certainly like to see us in that situation. And I do worry with respect to health equity that those underserved among us are getting less access to both the vaccine and appropriate testing, that they should be. So there still is whether you're looking locally within neighborhoods or whether you're looking worldwide, I think we have a long way to go to make sure that we are truly being equitable with respect to how we are handling patients in this regard. And it will come back to hurt us all, as Judd mentioned. We'll have other viral variants evolving somewhere. So, somehow we need to, as we all know, find a way to cooperate worldwide and have a system in place, whether it is for SARS or some other virus in the future. I think, we need to really grasp this reality and take appropriate steps, which is a very tall order, but fundamentally speaks to health equity and what we need to do for all of us, I think.
Adding to that, one point that Judd really did bring up was the fact that funding is an issue across the world for not just vaccines and testing, but sequencing. And so it's really hard to keep ahead of the variants when not everyone's able to sequence every sample. So variants can slip under the radar if we're not looking for them. And the only way to find variants is by sequencing. And sequencing is extremely expensive, requires specialized instrumentation, specialized staff and training. It's not something that everyone can do. And until we can have that broad surveillance network, it's going to be really hard to predict what the next variant is going to be, when it's going to happen, where it actually came from, and how to stop it. And that's even further down the line than just the vaccines, which people don't have access to. So it's hard to get worldwide sequencing surveillance when we can't even get worldwide vaccination. And so it's really like looking down that pipeline of every single step that needs to happen. It's really disheartening to kind of see we're not getting vaccinations across the globe. And so in the future, it'd be great if everyone gets vaccinated and we can do routine surveillance. But there's a lot of pieces that have to fall into place first. And I think Judd really highlighted that.
Yeah, I'll step off of that too, in terms of the sequencing inequities. And, we recently looked to see, by continent, how many sequences are deposited in the public databases from each continent. And the vast majority are from Europe and the United States, and then far fewer in other continents. For example, South America, fewer than 3% of the total sequences in the database are from the entire continent of South America. And for Africa, it's less than 1%. And as Judd showed in his presentation, we have variants that are emerging all over the world. And it may be, again, based on the data that he showed, it may be in places where there is less access to both vaccination, but also less access to healthcare, where it's possible that immunocompromised individuals have less access to treatment. And so they may be infected for longer periods of time, and there may be more of an opportunity for new variants to emerge within these populations. So it is, I think, very important for us to try to find ways to bring down the cost of sequencing and to distribute it more equitably around the world, so that we can have a surveillance network that truly works together to improve the healthcare around the world. We can't just sequence ourselves and here in the United States and think that we're doing a good job at protecting against infections, because they don't know borders. And as we see an infection that emerges somewhere in the world, it doesn't necessarily stay there. So we do need to improve our implementation of surveillance.
So thank you. And on that point and others just stated, based on Judd, your work, the monoclonal antibodies are less effective in Omicron, with Omicron variant. That's my understanding. And I don't know whether any of these new therapeutics are any more or less effective, but if you could comment on that, on the effect for monoclonal antibodies and therapeutics with the evolving genomics of the virus, and secondly, the health equity implications of this, because if therapeutics really are the future, if it's the new Tamiflu, is that really accessible to most people? My sense is there's limited stock, it's hard to access, driven by insurance coverage, but if you could address that as well.
Yeah, I mean, limited stocks and access to these different therapies are one part of it, but then it's made way more complicated by the fact that the correct therapeutic needs to now be matched with the correct variant. So as we saw with Omicron, many different monoclonal antibody therapies no longer are effective. So not only do you need access to them, you need access to the right ones, and you need to know which variant you have for it to truly be effective. And for all three of those things to be true, you truly have to have very good healthcare, which is just not the case everywhere. And what we've seen so far in the first couple of years of the pandemic is how rapidly the virus has been able to, gain resistance to monoclonal antibody therapies, beginning to build resistance to vaccines. What does that mean for the antivirals as those begin to come out? We've had remdesivir for a while. Luckily, we haven't seen resistance emerge to that. But how long, will it be before we begin to see, the one I can never pronounce. I'll call it MAL, resistance arise, or some of the protease inhibitors. We know that this happens. We've seen it with other viruses. So, treating those antivirals with, the proper stewardship is something we haven't even begun to think about, but it's going to have to happen sooner rather than later.
Thank you. Thank you. Great discussion. And we can come back to this health equity discussion as well, but I'd like to move on to the next discussion question, which is what are the implications of this work for dissemination and implementation science? Any thoughts, Egan from Northwestern or Karen here, but
yeah, I guess
it's evolving too. Yeah.
Right. I mean, I think I think it builds off of the discussion that we were having on healthcare equity, and that, I think we do, knowing what we know or what we think we know about how these variants emerge and in what sort of settings they emerge, actually being able to disseminate healthcare equitably and to making sure that that treatment is, especially within these immunocompromised populations that we think are at higher risk of developing new variants or evolving new variants. I think it is important that we are, that we target those populations for therapeutics, for effective therapeutics, for rapid therapeutics, and for vaccination, and that we are watching closely in terms of what their response to vaccination is, as we're seeing that it's now looking like there's going to need to be a fourth booster or a fourth vaccine for those that are immunocompromised, and making sure that those are disseminated, that those populations do receive those extra boosters, the extra vaccinations, I think will be important for reducing the risk of new variants emerging.
And in terms of healthcare dissemination too, it's one of the things that I'm struggling, and I continue to struggle to wrap my mind around is how quickly everything can change. And research and healthcare, as an enterprise, is not typically responsive on the order that this virus moves and changes. So how we are tracking these variants, how we're testing what their impact is, how we're disseminating that information, and then how we are implementing those policies, from basic science all the way into the clinic, to do that in the time period that we have to is a major challenge. How that implementation challenge can be overcome, I don't know. I think that with each variant, we're getting better. When we think about Omicron, when it emerged in South Africa, researchers there very rapidly reported it and put it on people's radar. That by the time it reached the United States, there were already several studies looking at which monoclonal antibody therapeutics may be effective and which ones might not be. And so we actually had a decent amount of fair warning to implement the proper policies by the time that it was here. Can we stay on the ball And can we do it even faster and more equitably across the globe? That's, I think, the million-dollar question.
Yeah, and so to that point, Paige, I wanted to ask you as well. Maybe you were about to say something, but I was going to say the implementation, like as you said, Judd, is not just into the hospital, into the clinic space, but it is, do we do sequencing for surveillance routinely, both in the human patients we have in our system, but also zoonotically? Is there, as a microbiologist, how would this happen to create a surveillance system?
So other institutions, especially more like research focused, have done wastewater surveillance, and that doesn't link it back to a patient, but what they do is they look at varying variants within wastewater. And that's more of a focus of research institutes not related to humans. At healthcare centers like our own, we do a broad surveillance. So here at North Shore, we sequence all COVID positives. However, that is not a fast turnaround time. So I say we sequence everything, we bank our positives, and we de-identify it's anonymous, and then we're able to sequence. And so we can say, okay, we see Omicron, we don't see Omicron. And so there's two key points there. One, we don't ever link it back to the patient or put it back in the chart. And so I think that there is a disconnect with the healthcare dissemination on that, because providers, arguably, they do want to know if their patient has Omicron versus Delta versus whatever else is out there. And that's just not the point of surveillance. Surveillance is to find out what's there so we can guide principles. So once we saw, 90% Omicron, that most patients are not going to respond to those treatments. And so that's what really the point of surveillance is, to say patient A has Omicron, patient B has Delta, is to say what overall we're seeing in the population. Other healthcare systems are looking at just the patients who are hospitalized and sequencing those, or just the outpatient or just pediatric, and focusing that way. And by doing that, it can skew the data. So if you're looking at the healthiest individuals who are out, who only just present because they want to go on vacation, you may see a more mild variant, or be skewing your data to include really what's just going around the general community versus what's making people very sick. So there's a lot of different ways to do the surveillance, whether it's wastewater or specific populations, or broadly like we do here. I believe Northwestern does the same thing with a very broad approach. There's really no uniform way of surveilling. And unfortunately, I'm hoping that as time goes on, we can get across the world a better understanding of who to sequence, when to sequence, and how to share these results in a timely manner. And I know a lot of places, including ourselves, have a 1-hour PCR test, and patients and providers are very used to getting their results in 1 hour. Sequencing is not a 1-hour test, and it will never be a 1-hour test. It takes days, it's extremely expensive. We don't just do one patient at a time, and I think that's important to emphasize as well. So while our testing can be very fast and you can get a result, you can do treatment, sequencing is more meant for surveillance.
Thank you very much. This could be its own seminar, this topic, because it is just so huge to think about how the innovation that would be involved with zoonotic sampling and where to start. There is a comment from the chat box, which is, how useful is antibody testing? Are patients developing different sets of antibodies depending on the type of variant they're infected with? Which might relate to this question of, should we test antibodies?
So right now we are testing antibodies, and the sophistication of the antibody testing is that, we have IgG or IgM. What that means, I don't know in the spectrum of a viral infection. We have some antibody tests available to us that will pick up spike protein that will be indicative of a good immune response to a vaccination. We have others that are going to detect the nucleocapsid, which will be indicative of a past infection, not vaccination. But we don't have antibody tests that are available that are going to discriminate Omicron from Delta, from all of the other variants. They're just not available. So, and we're not really even sure what to make of the antibody testing. Clearly it goes up and it comes down fairly quickly, if we're just measuring antibody levels quantitatively. But is there a cutoff beyond which people should get vaccinated, or what that all means? We're not entirely sure. I'm not an immunologist, but we get into not only the humoral but the cell-mediated response. It's quite complicated. So I don't think we have the tools nor the answers yet to use this in any other way than what we've been doing.
Thank you very much. I think we'll move on to the next discussion question, which is what are the implications of this work for policy?
And I'll just comment, maybe I'll start, but I think, you know, the discussion we've been having in the last few minutes really, you know, points to policy changes that are fundamentally needed for us to operate more quickly. I think we've all struggled with the slowness that we were able to roll out testing, get support. You know, a lot of the collaborations have been, you know, person to person, not mediated by any federal agency or even a professional group. You know, it's been Northwestern calling us and vice versa. So I think we really need to look at the infrastructure in this country and around the world and have policy that supports us being able to be more nimble, to share samples, to, you know, evolve testing as it's needed, and to provide, you know, what's needed to take care of people who are, you know, not in a wealthy country, not, you know, where they have ready access to, you know, the best of healthcare. I think it needs to be there for everyone. And that's going to take a lot of policy changes and also investment in our infrastructure.
Great answer. I was just wondering if there might be a new paradigm like after 9/11 with biodefense, where this really became a major investment everywhere. And yet, here we are. So
Here we are, yeah. I mean, there was investment, and there was activity after flu, looking at, you know, what we would do to, you know, be ready for the next pandemic. There were federal studies done and plans laid out, and they were abandoned and forgotten, unfortunately, budget cuts and so forth. So I think, you know, hopefully this will be in the end a wake-up call to how we need to position ourselves to be ready for whatever is to come.
Thank you very much. Yeah.
I can just really quickly jump on that and say that, you know, I know I sound like a broken record, but genomic surveillance, I think, is the thing that we really do need to build up. And it's something that I think was under-recognized, how important and how beneficial it could be to healthcare and public health and before this pandemic. And I think it's really been a star over the last couple of years in terms of what it's been able to do. And so my hope is that that is going to be broadly recognized by policymakers here in the United States and abroad, and that there will be a lot more effort and resources put into building a global network of genomic surveillance, not just for SARS-CoV-2, but for the other pathogens, the next pathogens that are out there, but also the other pandemics that are ongoing, like the ongoing pandemic of antimicrobial resistance. I think that genomic surveillance has a huge role to play in that, and I think it's been under-recognized, and hopefully over, you know, going forward, we'll have more effort put into that.
Absolutely. Other thoughts on policy?
Yeah, just to touch, you know, we had touched about this, the administrative kind of burdens to get this kind of work done, even within the city of Chicago, trying to work between healthcare institutions without there being an overriding and guiding kind of body. And it becomes even more complicated then when we try to implement that type of approach across the globe. We are the center that I work in with Egan is located in the Institute for Global Health. And so we try to partner with institutions to do two things. You know, one, provides surveillance capacity in real time, but then also to help build capacity on their end so that they can begin sequencing on their side. But that takes time and that takes investment. And the simplest little things end up taking the most amount of time. Just shipping a package from here to Nigeria or from Nigeria to here is hours and hours and hours of dedicated time listening to, you know, couriers' ringtones. It's just, there are small things end up making a big difference. And I think if these small hurdles could be broken down, then we'll start seeing more and more of it. But
well, you bring up Nigeria is an emerging economy that's actually quite impressive, and I wonder if the thinking about this in terms of capitalism or just what drives things to happen. Are there ways that internationalizing this, democratizing it, could actually be profitable enough to motivate governments and big corporations to see this as markets. If I can say that, just that seems to make the world go around. Is there money in this enough to motivate non-government entities to globalize it the way that we have with the internet and with smartphones and this kind of thing, the testing, the surveillance? And is that a policy question, or is it more of a translational question?
I don't think there's ever been a whole lot of profit in infectious disease, unfortunately. So I think it's a huge public health and medical question. I think that, infectious diseases are one of the major killers around the world, especially in low and middle income countries. But it doesn't tend to make a lot of money for people. So I think that would be it would take some very creative thinking to find ways to monetize that and make it, but also to make it so that it's not profiting too much or on individuals, especially in low and middle income countries.
Yeah, I agree. I think diagnostics in general is not a profit maker, and in most places the policies that we encounter are pretty much racing to the bottom in terms of reimbursement and finding ways to cut costs, and I think there's false economy in that you're really undermining our ability to appropriately diagnose patients. And in ID, I think it goes beyond the diagnostics. I think it's also treatment. That's why we have a dearth of antibiotics in the pipeline for drug-resistant organisms. So again, some policy examination might be in order for all of this.
I also want to emphasize that it's important. Oh, I'm sorry. Oh,
No, please.
As important as funding is, like, we definitely need funding across the board. There's a lot more that goes into it, like trained personnel, and that's something that can't be easily replaced. And for all the healthcare systems that are performing sequencing, we're still performing clinical tests. So for cancer, for all other infectious diseases, anything blood-related, hematology, we're doing it all. And so it's really hard to come up with just sequencing and a crew just for sequencing, instrumentation just for sequencing, supplies just for sequencing. There's a lot more moving parts. Even if you have the money, when we went through a shortage across the country of plastic for months, it was hard to even get pipette tips to even do testing for normal conditions or cancer or infectious disease. And so there's so many moving pieces. Money really, really helps and it's important. We also have to keep in mind that there's people behind this who are actually performing the testing, the reagents that we need. And so it's not quite as simple as being able to throw money at it and walk away. Money is 100% required, but there's still so much more that goes into it.
Thank you all. I'm going to move on to the next discussion question, which are what are the future translational research implications of this work? And if you could also comment on studying the sociome as part of surveillance to help us better position every community for testing and treatment, that would be an interesting discussion as well. So to any of you.
Just to quickly start us off, I think one of the things that we're all really anxious to see is exactly what the periodicity of this virus is going to look like. One of the things that is required for a virus to truly be considered endemic is that we get predictable waves that come on a seasonal pattern or variation that occurs in a predictable way. And we are not there yet, as Omicron clearly showed, but some aspects of the pattern are beginning to emerge. So continuing to follow up on this type of research so we can establish what we can expect at least puts us on a firm grounding. So it's not like every new variant is like, oh my gosh, where did that come from? But then the second thing that I just want to note is, as the population does become more and more protected, I think it's going to be vital that we're communicating properly, that it's not that the virus is becoming less virulent per se, but that we are better at protecting ourselves. Because it's a very different message in saying the virus is becoming weaker versus we are making ourselves stronger. And I think really discerning the difference there quantifying it and being able to message it is going to be really important.
So, yeah, thank you. And you talked about the kind of natural design of the spike protein going back to the start of your talk, which is fascinating because it's so diabolically naturally brilliant. This design, it's almost like a space age robotic weapon of some kind. It's just unbelievable. Is understanding that the biological mechanisms going to lead to druggable targets to perhaps in the future, the way the mRNA technology made these vaccines possible? Are others learning things here that might be transformative in drug design?
Yeah, I mean, absolutely. Every bit of basic biology that we learn about this virus is a weapon that we can use. This virus has a genome of 30 kb long, and it makes 20, 30 plus different proteins. And all of our therapeutics are really laser focused on spike. And then we have one against the polymerase, maybe two, and now we have another one coming out against the protease. And that's about it. There's a huge number of genes in this virus that do essential functions for its replication that we really haven't even begun to explore the therapeutic potential behind. So understanding the mechanism of spike, which we really had focused on, and which I really elaborated here in this talk, has been incredibly informative for us to understand exactly what mutations are arising, why they're important, how we can develop new and better monoclonal antibodies. But understanding the rest of the virus replication cycle is going to be equally an important contribution to that.
So where should we invest the most in the future in translation science? Or which areas would you like to see develop the most? I've heard a lot of comments about surveillance so that we can basically have a radar and not be blindsided like we were in 2020. That sounds complex because it's pretty much, it's ecological, isn't it? It's not only humans, but also zoonotic species, it's wastewater, what would that look like? And I know there's only a few minutes left. What designs would be possible to get us to a point where we had a surveillance system? And I'm asking, Judd, I know that you're involved with this work, and others probably here too are as well. Can you talk a little bit about what surveillance looks like, and then we can conclude very shortly?
Well, I mean, I think it's a great question, and I wish I had a perfect answer for you. Right now, one thing that I think we can focus on fixing is there's a huge inequity in how we're performing our surveillance. We are very oversampling particular regions based on who happens to have the funds or the expertise, and we happen to be dramatically undersampling other regions. This is a global pandemic. The virus is everywhere, and it's continuing to spread and evolve everywhere. So coming up with a way that we can normalize the sequencing to be a little bit more equitable across different regions, I think is a huge step forward. And that in and of itself is easier said than done.
I can jump in real quick and just say, I mean, we're definitely a lot more primed to do this now than we were 10 years ago. For example, the technology has advanced quite a bit in the last decade to do genomic surveillance. And it's just, I think it's going to continue to become cheaper and easier and more portable and easier to perform. And so I think probably those would be the places that I would recommend investing in, is just to find ways to make that genomic surveillance even cheaper, even easier, and more wide, so that it can be performed more widely and more readily in places that don't necessarily have the resources that a European or a United States or a US research institution does. That it can be performed in labs, in clinical labs, that it can be performed in places that don't have access to full molecular biology labs. And I think we are moving that direction, and we're certainly moving there even faster now than we were even a few years ago. So I think it could be on the horizon. It's not we're not there today, but it's a direction we should be going.
I agree. I think the folks who are doing this now are those who got grants to do it. This is not supported by any federal mechanism. So it's either research grants or philanthropy or something. So that creates a very uneven pattern of where this is being done. And it's in the wealthier areas, as we've talked about, and those sorts of things. It's interesting because the public health system rolling up to the CDC is completely separate from the hospital systems. And many of us have been advocating to try and bring them together in some way, because really the hospitals are on the front lines of public health. We're seeing the patients in the EDs. Yes, we'll send them off to, samples off to the CDC, but they're not equipped to be doing testing on the scale that we all need. Neither is the Illinois Department of Public Health or any of our state societies. So somehow we have to create a network that is supported by funds other than just grants. That will stand ready to do the appropriate testing, not just locally, but testing from elsewhere. It's crazy that Judd's lab has to go through what they have to go through to send a sample or receive a sample from Nigeria. We somehow need to come up with a network, a global health network that will facilitate all of this, so that we can be ready. And that gets back to policy again.
Very well said. I think on that remark, I think we will come to an adjournment of this seminar, which has been just absolutely fascinating and brilliant. Thank you all so very much. And perhaps we can invite you back when the Global Health Network is built with grant funding that I'm sure will be around very soon. But I'd just like to take this moment and thank you, each one of you, for your major contributions to this transdisciplinary panel and to really a fascinating presentation. Thank you so much. Thank you, Paige, Egan, and Karen, for joining us today. And I think there are a number of points that came out of this. There's the evolutionary process is complex, really extraordinary, and has been fairly well understood. I would think it's fair to say, which has informed a lot of surveillance and potential interventions. But the news is not necessarily that Omicron is just less dangerous and we should all kind of take our masks off and relax. It's clear it's more complex than that, and that the booster works with Omicron, definitely. Without it, I think we're all at risk. And so there's a lot of work left to be done, a lot of inequities to try to address, but there's also been evolution scientifically, which has brought us to this point, which has been very encouraging. And perhaps we could get to that more of integration with public health and medicine so that we can better surveil the ecosystems and be less prone to being caught off guard like we were in 2020. So thank you very, very much indeed. And if you could advance the slide, Rachel, that would be good. Great. Well, okay, so just to finish up here, thank you all participants for joining us today. For questions, comments, concerns, or requests to present at a future seminar, please contact me. My email is listed on the slide. And if you could all please take a brief survey regarding today's presentation. It should be in the chat or will be shortly. And you'll also be emailed a link. And this is an evaluation of the seminar that we do read and we value, and it is used to make this seminar even better. And then our seminar series will be back next month on March 15th, where we're going to be welcoming Dr. Roy Perlis from Harvard Medical School, who will be presenting some of the research from the COVID States Project, which is a large public survey research program that is tracking COVID-19 public perception, including misinformation. So we'll have a panel, including some members of the community, talking about vaccine misinformation next month. So lastly, the recording of today's webinar will be distributed shortly, and it will be on our North Shore YouTube channel. So for any of your colleagues, any learners, please feel free to distribute this. I think this is a particularly special and informative seminar today that I think would be great for residents and students to see. especially if they missed Judd's grand rounds this morning. But I think it got into a lot of much broader issues too that I really appreciated your comments about. And so we will send a link to that. And just thank you all and have a really great rest of the week, and please stay safe. And go home and root for the Americans in the Olympics. All right. Thank you all so much.
Thank you, Sean. Nice job, Judd.
It was really nice, Jen.
Thank you.
Thank you.